Okay. Yeah, it's okay. We can actually start today's seminar. I think you gather some international also newcomers to this seminar, so this is very good. Today is the time of Dr. Viviana Monje-Galván, originally from Bolivia, La Paz, right?
Yes,
If I'm not mistaken, is the capital with the highest altitude on Earth. And
I
now,
think so.
now currently you are in New York, which actually gives you only 10 meters above the sea level. So
Yeah,
you
it's
you did
nothing.
some giant leap there.
Mm.
Okay, so you are she's a group leader and also an assistant professor at the Department of Chemical and Biomolecular Engineering at the University at Buffalo. She's been studying basically cell membrane modeling and biomolecular interactions with applications in different diseases, especially in viral infections, cell death, cancer, just to name a few. Interestingly, so you have worked also with SARS-CoV-2. That is actually an interesting activity that we are trying to develop with Fernando. And today's talk is going to be about lessons learned from lipid membrane studies. So the floor is yours, Viviana.
Thank you, Adolfo. Good morning, good afternoon, good evening, depending where you are in the world. I'm learning to say all the three whenever I start a talk online because we're in so many different places, but it's very exciting that we can share our passion for science, we can learn from one another, and it's my privilege to share the work of my group with you this morning. As Adolfo was saying, I started doing membrane modeling in grad school at the University of Maryland. And I have two slides on my very first work on membrane modeling, which actually started all my love for molecular dynamics and studies of membranes and how one of the strong suits from my labs come from. So I will share again those two slides from my very first studies, and then I'll focus on two examples of simulations that we are currently studying in my group. And I named this talk Lessons Learned from Lipid Membrane Studies because sure, we're after characterizing protein-lipid interactions, at least in the studies that I will show you today. But along the way, both in terms of the membrane size and the membrane composition, there are things where that I have learned in the past 10, 12 years that I've been doing this type of studies, and that we continue to explore in future research, right? I think most of the people who are in the call are familiar with molecular modeling. So I have just one slide with our famous equations from molecular dynamics, but that's it. I will not go so much in the technique, but rather focus on the applications we can look at. Are my slides moving? Do you see the new one with a bunch of pictures?
Yes, moving.
Okay. So very, very briefly, when I moved from Bolivia to the US, I came to the area of Washington, D.C., which is actually this tiny star at the bottom right corner, next to the picture of my two kids and the rest of my group. So my two kids are really the bosses of the group. They let me do all the work that I do with my students, and without the support of my family, really any of the research that I do wouldn't be possible. We lived in Maryland for about 10 years. I did my undergraduate and graduate studies in chemical engineering at the University of Maryland. Then I moved to Chicago, which would be actually behind this picture of the group with all the flags. I spent 3.5 years there and a group of Gregory Both, and there is where I was studying more viral dynamics. And when the pandemic hit, the COVID pandemic hit, we basically all shifted from the proteins we were studying at that time in HIV to the corresponding proteins in SARS-CoV-2. And I was, of course, doing protein membrane interactions. And then about 2.5 years ago, I moved to Buffalo. Right here, you can see a picture of the university with our mascot, with the face of the buffalo. This is the North Campus where the engineering school is. One of the most famous views from this area is, of course, Niagara Falls in the top. And then the very top right corner, it's a view from the airplane from Buffalo downtown. It's a very nice place. You see Buffalo here at the bottom, and then in the top most corner, that's Canada already. So we have a bunch of bridges between Canada and the US, and we're right at the frontier. We're very fortunate to have a supercomputer here at the university, the Center for Computational Research, or CCR. And I've been very, very fortunate to have an amazing team of students. I currently have 3 PhDs. I work with one of my master's students, and then a few undergrads who have been very committed. For being undergrads, they have been very good in making progress in all the projects. And later in the next slides, I will share with you 2 projects, one of them that is already published. Actually, that's how I got a chance to interact a bit more with Adolfo in the special issue that he was coordinating in Frontiers in Chemistry. And then the other study that I will share today, we just submitted and we're working on the review, so hopefully it will be soon published as well. If I were to show you just one slide from my group, this would be it. So we basically tried to look at the role of lipids in different mechanisms of disease using molecular dynamic simulations. We focus in all atom molecular dynamics, and then we're also doing some enhanced sampling techniques. So, so far we're not working with coarse graining, but we may need to do that depending on some protein assembly studies that we're starting to look at. But mostly we do all atom molecular dynamics. The strength that we have is that we take quite a bit of time and we spend some effort to come up with realistic membrane models. And by realistic membrane models, what I mean is that every different organism has a particular lipid distribution in their cell membranes, and even different compartments in the cell have different lipid distribution. So if you want to study a process, let's say in the plasma membrane versus a process in the endoplasmic reticulum, in my lab, we do the best effort that we can to find the most characteristic lipid species in these different organelles for the specific organism we're studying. So whether it is yeast cells or mammalian cells or cancer cells, for different type of cancers as well, we try to look for lipidomic studies, or we work with experimental groups to be able to reproduce this membrane model, or reproduce in the membrane model the lipid diversity, but mainly the mechanical and structural properties of this membrane environment, so that then we can study the interaction of other biomolecules with this membrane, right? It can be protein lipid interactions, they can be on the membrane surface, they can be transmembrane proteins. And actually a project that started as a small project has turned out to be 1/3 of the research we do in my lab now, and is the interaction of small molecules with membranes. So I don't have the study for that in the presentation for today, but I'll briefly tell you that we are looking at organic molecules that have the backbone very similar to cholesterol, and we're trying to understand how they interact with the membrane in the context of antibiotics or creating pores in the membranes and membrane permeability for repurposing these naturally occurring molecules as potential drug treatments. So it's been a fun study, and one of my students has taken this study really to the next level, starting to look at enhanced sampling and maybe trying to do very little method development in the sense of selecting smart collective variables for smart sampling. So that's that for the small molecules. And then today what I will show you is a couple of studies we are currently doing of protein lipid interactions. Just a shameless publicity at the bottom, you can see different organizations that I volunteer with. So I work with the Society of Biophysics in Latin America, or SOBLA. I'm very much involved with Latinx in Chemie, in chemical engineering. At the university, we have a program called WISE, Women in Science and Engineering, and the LSM program, which is a program that supports and promotes the participations of minorities in science. I was actually a scholar as an undergrad and a graduate student of this program in Maryland, and it's my way of giving back. I also serve as mentor for students participating in this program at the University of Buffalo. And if you want to read a bit more about the team, the students, and the projects we have, this QR code in the bottom left corner will take you to our website, and I have it in the very last slide also. So very briefly, I mentioned that all our studies are centered or start with realistic membrane modeling. Cell membranes are mainly made by lipid molecules, or they create this sandwich, this bilayer, because of their amphiphilic nature. They have a hydrophilic head group and a hydrophobic tail. If you imagine them as a Lego set, you can create infinity of options between the different lipid head groups and lipid tails that are available in the cells. Mammalian cells have easily thousands of lipid species in their lipidome. Yeast cells, which tend to be used as model organisms for eukaryotic cells, have quote unquote only hundreds of lipid species in their lipidome. So sometimes it's easier to use yeast as a model organism to model the cell membranes of mammalian cells. And that was actually my very first study that I will share with you in a couple of slides. Lipids can have very small head groups, like the ones you see at the bottom of the screen. So you can have PA, PE, you can start to have more complex head group structures. I'll probably change to a laser pointer. There you go. So you can see this at the bottom right corner. You have these cyclic compounds in the lipid head groups. Those are inositol groups that can have phosphates. They can further have other sugar groups attached to the molecule. And those are these hexagon figures that you see in the illustration at the top of the slide. So the topology of the membrane surface can be very diverse, but also the interior of the membrane. So you can have very short lipid tails or very short fatty acid tails that can be 12 or 14 carbons long. Typically in mammalian cells or eukaryotic cells, you have a distribution of 16 to 18 carbons in length for these fatty acids. But we also have in certain organisms, in certain organelles in the cell, we have very long tail fatty acids, which can be 24 or 26 carbons in length. And all this diversity results in very interesting combinations. And if you put these different lipid species in different ratios in the membrane, then you have sections with different mechanical and structural properties. And you need that, you need this platform with this variety, this diversity of structure and mechanical properties for the cell to function properly, right? Depending on what mechanical and structural properties you have in one region, then certain type of proteins will be embedded inside that membrane. Other type of proteins will like to interact with it on the surface. And now we know that lipids are very much active in different processes in the cell, especially in signaling cascades. So that's where the motivation comes from my group to study protein-lipid interactions and lipid-lipid interactions, trying to mimic as closely as possible the environment we're trying to study in the cell. It's not passing the slide. Okay. So I mentioned that I was going to share 2 slides with you from my early work and what sort of got me into membrane biophysics. And it was actually creating complex membrane models for yeast, again, as a model organism for eukaryotic cells. We did a thorough literature review going back to studies from 1999 of different lipids that are present in the endoplasmic reticulum, transgolgi network, and plasma membrane of yeast Saccharomyces cerevisiae. The main difference was, of course, the content of cholesterol, which are the yellow molecules in the figures you see on the slides. Very low content for the ER, less than 5%, 10 to 15% for the transgolgi network, and then over 30% for the plasma membrane. You can see the diversity of lipid head groups in the bar plot that you see on the top. Phosphatidic acid, phosphatidylcholine, phosphatidylethanolamine, inositol lipids, serine, and for yeast, the major sterol representative is ergosterol instead of cholesterol. We learned from these studies that even the chemical structure of these lipids, so replacing ergosterol by cholesterol, does result in a change on the mechanical properties of the membrane model in simulation. We looked at membrane thickness, we looked at the electron density profile, we looked at the order parameters, so structural parameters of the membrane, looking at the distribution of the angles between the carbons and their respective hydrogens down the tail of the fatty acid to get a metric of the order or the organization on the interior of the membrane. This order parameter is a value between 0 and 0.54. The highest this number, it means that your we say the bilayer is more ordered, so the tails are more parallelly aligned to one another. The lower this value, the more disordered the environment. The major conclusion we had was for the endoplasmic reticulum and Golgi network, the blue and red dots in this plot, the order was much lower because they had lower cholesterol. And we saw that our models were reproducing the higher order in the plasma membrane, the black dots in this plot, because of what is known as the cholesterol condensation effect, right? You have more cholesterol in a bilayer, your lipid tails are more parallelly aligned to one another, they are tightly packed, and the thickness, the membrane becomes thicker or bulkier. So that's known as the condensation effect. We also looked at how the lipids were rotating, not only with respect to their main axis, but what is called the lipid wobble. So if you have a lipid and you have a head group right here, this is the neck region where the glycerol backbone is. If the glycerol backbone stays put in one place, imagine a wooden spoon and you're cooking something, so that's the motion, right? That's a wobble motion. It's a rotation or this conical rotation with respect to the glycerol backbone. And we, from the simulation, because we get such a large amount of data, we were able to get a time correlation and the decay of the time constant for this wobble rotation. And we found out again that when you have more cholesterol, this rotation, the time scale for this rotation is much slower than when you have less cholesterol and the membrane is more fluid. So this was my very first study in membrane modeling. I really enjoyed it. I learned a lot about molecular dynamics. I was studying with Jeff Kloda, who has been a great mentor until today. I'm very fortunate to call him my mentor and hopefully also friend. And after we published this work, we started to study protein-lipid interactions with it, and then we did other membrane modeling studies. So based on that, I decided that in my lab, I was going to do also membrane modeling, realistic membrane modeling. Typically for people who do membrane simulations, you know that by far membranes are usually pure bilayers or maybe ternary mixtures because that gives you enough of a headache with the analysis. In my lab, we have our membrane models that range between 5 and 9 different lipid species. And this is again to reproduce the mechanical and structural environment of the membrane we're trying to model. And we have proven that if you have different lipid distributions or different lipid ratios and different lipid species in the model, you indeed can capture different interactions of other molecules with the membrane. So in terms of simulating systems, again, very brief, a flash overview, because I believe all of us are somewhat related to simulations. We can do modeling at different scales, depending what we're trying to learn about a system. We can do different types of simulation techniques. We can do quantum mechanics simulations for very small system, a few tens or hundreds of atoms. If we're more interested about how molecules are interacting with one another, then we get into the regime length scale at time scale of molecular dynamics or Monte Carlo. And as we try to learn more assembly of larger protein bodies or membrane protein interactions of a larger scale, then we start to use coarse grain or ultra coarse grain molecular dynamics. And if we start to lose the resolution of organelles or atoms or molecules, we maybe get into phenomenological models or mathematical modeling to understand how, for example, how cells can diffuse, let's say, a tumor, how the cells from a tumor can diffuse in tissue and all of that. So different time scales, length scales, different simulation approaches. And as I mentioned at the beginning, my lab focuses in all atom molecular dynamics. There are many different molecular dynamics engines and different software packages. We use the ones at the bottom, so GROMACS or NAMD, depending on the size of the system, to improve the performance, the nanoseconds per day. We use VMD to visualize our system and run some analysis. And we use Python libraries like MD Analysis, MD Trash, or in-house scripts to do the analysis of our simulations. So for molecular dynamics, what we're essentially doing is starting from a system, we get the coordinates, whether from chemical structure or crystallography, cryo-EM, depending for the data available for given molecules, for proteins or small molecules. We solved Newton's law of motion with this force field or set of simulation parameters to model the bonded and non-bonded interactions between our atoms. And we essentially generate a movie of how the atoms in our systems move based on the other atoms in the vicinity of them. Right? We send them to run on a computer cluster, and then we do the analysis. Now, membrane systems, if you have on the smaller end of a membrane-only system, you can have anywhere between 80,000 or 100,000 atoms. Those are very small membrane patches. And you can go very easily all the way to 2 million atoms for membrane protein systems, right? At the all atom scale. Of course, you can go larger, but that's the typical range between 100,000 and 2 million atoms. For each of those atoms, you're solving 6 equations at a time, 3 for position, 3 for velocities. So without the use of high performance computing clusters, for us, the job would be really impossible. We've been fortunate to have time allocations in the Anton machine, which is a machine designed by D.E. Shaw that is maintained by the Pittsburgh Supercomputing Center. We apply for time every year, and we've been fortunate to get some time there for the long trajectories we run. We used to have resources in Exceed. We'll be applying to the new program called Access. And then we have the CCR, which is the Supercomputing Center from the university. Now, why do we need, or why is it for us beneficial to have access to, for example, a machine like Anton, is because our membrane models, I mentioned before, they have between 5 and 9 different lipid species. One of the major challenges for us is to ensure that we have enough sampling of the lateral diffusion of these lipids. So while typical membrane simulations will run for 300 or 500 nanoseconds, we typically run ours for at least 1 microsecond or 2 microseconds, especially if we're trying to understand how this membrane interacts with other biomolecules and what is the fingerprint of that interaction between a given protein and the membrane. So we run very long simulations. With the computational resources today, we can certainly get our trajectories within the lifetime of a PhD student or of a master's student, but having access to Anton or other very fast type performance computing center is definitely beneficial for us. So the major hub that we have in my lab is understanding molecular mechanisms of disease using all atom molecular dynamics. Using this type of simulations, what we're basically trying to do is understanding these microscale interactions and give potential explanations to what we can observe in experiment. We also use it not only as we use these simulations not only as a computational microscope, but because of the underlying nature of the simulations, which are based on statistical mechanics, and we have the simulation parameters for the interaction between atoms from quantum mechanics and empirical force field development. We also try to use it as a predictive tool. So if we don't have an experiment that will be looking at a specific protein lipid interaction, we try to make predictions of what may be happening at this very small scale, atomistic scale, and then hopefully inspire other groups to do the experiments to test that hypothesis or help them identify experimental targets. So we use the simulations from both ends as a computational microscope, as well as a tool to predict interactions between biomolecules. So we start from a membrane model. Again, depending on what is the particular disease we're trying to understand or the specific organism we're trying to understand, we'll go on the literature or work with our collaborators. I'm very fortunate to have 3 experimental collaborators, one for the small molecules project, the other one for the study that I will show you today, the first study that I will share with you, which is in the context of mechanisms of cell death. And then another collaborator that is looking at lipid droplets and membrane properties in that end. So with the information they can provide us, we come up with a membrane model. We run the simulation. I'm showing this picture here because this particular membrane model had a lot of sphingomyelin, and it started from a liquid disorder phase. You can see the tails very much entangled, and after the simulation, you see this very nicely and parallel fatty acid tails inside the membrane. So that was very exciting to see. This was a high cholesterol, high sphingomyelin content membrane, and you can clearly see the ordering of the lipid tails as the simulation progressed. Once we have the membrane model all settled, we start to look at the interaction of different molecules with the membrane. Most of our projects are with peripheral membrane proteins or with molecules that are outside of the membrane, and we're trying to understand how they cross the membrane. So these biomolecule interactions with the lipid head groups is something that we're very much interested in characterizing. And this is something we're calling, and other groups are also calling, the fingerprint of this particular interaction. So in the case of proteins, if we have a protein interacting with the membrane, we look at that red region that you see in this diagram at the bottom of the slide. This is the vicinity of the protein. We measure the mechanical and structural properties of the membrane right underneath where that molecule is bound. Then we define a buffer region, and then we compare the mechanical properties very near to the protein versus the properties of the membrane in bulk. And then we try to determine what are the things, how are the things changing, how is the local lipid environment changing because of that protein lipid interaction, and how that affects the mechanical properties of the membrane. And then we look at membrane properties such as the order parameter, the lipid distribution underneath where the protein is. We also look at hydrogen bonding. We look at radial distribution functions to see how the lateral organization of the lipid is changing. One of the challenging things we're trying to also examine is whether the top leaflet and bottom leaflet are coupled or not in their response to the interaction of another molecule interacting with the bilayer. So definitely very exciting field, and I think a lot of people are starting to look at this because, again, now we know that lipids are very much active participants in different cellular processes. So the two cases that I will share with you today are one from a native protein in the cell. It's called the MLKL protein. I will talk a little bit more about it in a minute. This is in the context, again, of mechanisms of cell death, protein lipid interactions of a protein that is known to help in the permeabilization of the membrane as part of a mechanism of cell death called necroptosis. I'll talk again about it in a minute. So that's one example of the studies we do of a protein that already exists in the cell and how it interacts with the membrane. The second example is from a viral protein. This is a protein from hepatitis C. We're not really looking at viral dynamics, but once I get to this project, I will explain why we have an interest in this particular protein from hepatitis C. In any case, the second example is of a non-native protein and how it disrupts the membrane environment. In the first case, we're looking at the plasma membrane and how it interacts with a protein. In the second case is the membrane from the endoplasmic reticulum and a viral protein. So the first one, as I was saying a few moments ago, excuse me, it's a protein lipid interactions and membrane disruption in the case of a mechanism of cell death. This is of interest for diseases like cancer or neurodegenerative diseases where inflammation is a big thing. In many cases in this type of diseases, the cells can avoid apoptosis. And then another route that the cell has to destroy itself is another pathway that is called necroptosis. Apoptosis and necroptosis, they both share the first initial cascade of events, right? There's a transcription factor that gets activated, and there exists a bunch of signaling cascade events that end up in the phosphorylation of RIP kinases. It's a family of proteins that, in the absence of caspase, end up phosphorylating MLKL protein. And this is the last step of this cell death pathway. This pseudokinase, mixed lineage kinase like protein, is known to then be translocated to the plasma membrane and help in the permeabilization of the membrane. There is leakage of the cell content, the cell dies, which is good in the case of cancer cells, right? You want to kill the cancer cell, but as the leakage from the inside of the cell is present on the outside, there's an inflammatory response. And this inflammatory response can actually be promoting the formation of new cancer cells. So they know necroptosis is known as this double-edged sword for cancer treatments. while it kills the cancer cell, it results in inflammation that is actually an oncogenic environment. So what we're trying to do is learn more about this process so that it can be eventually leveraged in treatments to kill cancer cells, but prevent the formation or the proliferation of new cancer cells, right? The development of new cancer cells. Our question, or the question that my students are trying to answer, this was a master's student, Laura, and Antoine and Ricardo are still working in this project, was looking at, with our collaborators in the chemistry department here at UB, look at the interaction of MLKL with the membrane. We know it's the last step in necroptosis. It's known from experiment that it gets phosphorylated. It is now known that it also gets acylated, but there's still a big question mark in terms of how it permeabilizes the plasma membrane. Some people suggest that it needs to be a tetramer. Other groups said maybe a trimer. Other groups mentioned, no, it's only the phosphorylation step that is needed. More lately, our collaborators are showing that it's a different type of mutations that are helping the permeabilization of the plasma membrane. So we're trying to provide insight from all atom simulations of what domains of this protein are interacting with the membrane and how the membrane responds to that interaction, right? As a long-term goal, we want to see the interaction of multiple protein units with the membrane. The study I will show you today is just one protein in the vicinity of the membrane, and then we let the simulation run to try and understand which one of the 3 domains of the protein interact with the membrane and how the lipid distribution changes in that very local environment. So based on the experimental studies from our collaborators, we designed a 5 lipid species model for the plasma membrane. It is, of course, an oversimplification. This was a symmetric model because we have to start somewhere, but eventually we want to move to an asymmetric model and then start including sphingomyelin lipids and phosphatidylserine lipids, which are also very important in different leaflets of the plasma membrane. So this study was run on Anton, most of the simulations, 4 different replicas, a single unit of the MLKL protein with the membrane model. We started in different orientations, which is sort of a naive way to try to avoid bias in the simulations. Actually, one of the things that my student Ricardo is trying to do is find a better way to determine these initial orientations of the protein. Which ones are worth trying out of the many different orientations that you could position this protein near the membrane? How can we make this selection more efficient in terms of sampling different events in the simulation? So anyways, the MLKL protein has 3 different domains, red, blue, and black. The black one is the pseudokinase, is more of like a globular protein. The red one is known as the brace. So that one serves sort of like a hinge between the blue and black domains. And the blue domain is the 4 helical bundle, known in the literature as the killer bundle, because it's the one that is supposed to interact with the membrane and help with the membrane permeabilization and effectively killing the cell. So what we saw was in these 4 different replicas, the protein always binds the membrane. We found 3 times that the pseudokinase domain, the black domain, interacts with the membrane. The brace only interacted 2 times with the membrane. And the 4 helical bundle also 2 instances out of the 4. The most interesting bound conformations based on the feedback we got from our experimental collaborators were replica 2 and 3, so the 2 middle ones, where we have in fact the 4 helical bundle bound to the membrane, and then one of them was horizontal, the other one was vertical. The bar plots that you see on the slides are simply the frequency of contact between different amino acids in the different domains, color-coded in the same colors as in the snapshots. And you see that whenever the pseudokinase domain, the black domain, is interacting with the membrane, there are, if it's in the vertical conformation, right, like replica 1 and replica 4, sort of the same group of amino acids are interacting, maybe at different frequencies, but basically the same portions of the pseudokinase domain are interacting with the membrane. In a similar way, when the 4 helical bundle, the blue domain, interacts with the membrane, again, the same subset of amino acids are the ones interacting with with the bilayer. To us, it was very surprising to see this second replica, the behavior of this second replica, because the initial bound conformation was similar to replica 3, in which the 4 helical bundle interacted with the bilayer. And then midway our simulation, we see the protein start to lay flat, and the black domain, the pseudokinase domain, interact with the membrane. We did not see that in replica 1 or replica 3, which again bounded vertically with the membrane. What you cannot see in the current representation of this protein in the slide is that this red domain, the brace, is in different orientations with respect to the membrane. Our hypothesis is that in the case of replica 2, in this particular bound orientation, where the 4 helical bundle was initially bound in a vertical conformation, this brace region was able to actually create the hinge movement, and that's what allowed the protein to then lay flat on the membrane. One of the things that we were looking at, as I said before, was the fingerprint of this interaction, the fingerprint that is left on the membrane in terms of local lipid composition and change of mechanical properties in that local environment where the protein is bound. What you see now on the slides are the initial and final protein lipid contacts. So these are just heat maps between different regions of the protein, right? The pseudokinase domain, the brace, and the 4 helical bundle with respect to each one of the lipid species in our model. So what you see basically is when you have the pseudokinase domain interacting with the membrane, you have a certain ratio of lipids, versus when the 4 helical bundle interacts with the membrane, you have a different distribution. Other type of lipids are recruited or displaced from right underneath the protein. And that's exactly what we were trying to show. If the protein binds in different conformations, then there is a different redistribution, a different resorting of the lipids right underneath of it. In agreement to our experimental collaborators, what we see is that there's an enrichment of PIP lipids, where you see the brighter colors or closer to yellow. That's where you see enrichment of this type of lipids in that particular leaflet. The bottom row is the binding leaflet. The top row is the non-binding leaflet. You'll see that there are some differences. Right where the protein binds, DOPC gets displaced away from where the protein is bound, of course, in the binding leaflet, which is not reproduced on the other side of the membrane. You get a little bit of enrichment of DOPE. You get PIP1 and PIP2 lipids, which are inositol with phosphates. This is something that our experimental collaborators were also seeing, that inositol lipids are indeed recruited to where the MLKL protein is. And then we saw a marked enrichment of cholesterol at that point as well. We did look at hydrogen bonding, at how lipid packing defects change on the surface, and you can read that in the publication. But the main takeaway message from this first study that I want you to remember is that protein membrane interactions do in fact result in a particular fingerprint on the membrane after the protein binds, which I forgot to say in our simulations of 2 microseconds, the protein bound within the first 500. And then as you let the simulation run and the membrane re-equilibrate after this protein binding event, then we do end up with a different lipid fingerprint right underneath the protein, depending on the protein domain that interacts with the membrane. The 4 helical bundle, which in experiment is known as the killer bundle because it's believed to be in charge of the permeabilization of the membrane, in our simulations is the protein domain that can insert inside the membrane past the phosphate groups of the lipids, which are shown in orange tiny spheres on the slides. PIP lipids or inositol lipids and cholesterol are notoriously recruited to the protein binding site. And something that we're now starting to look at is how the cholesterol flip-flops between the leaflets in these studies. And we see that in all cases, the cholesterol molecules, as the protein binds the membrane, the cholesterol molecules are displaced and go to the opposite leaflet, so the non-binding leaflet. So there is some interesting response that we're trying to quantify as well. So that's for the first study. This again was a protein that already is present in our cells. And the second case study that I wanted to share with you today in the next few minutes is that of a non-native protein. In this case, it's a viral protein from hepatitis C, and how it interacts with different membrane models. So in this case, what we're trying to understand is the specific relevance of phosphatidylserine lipids in the binding and interactions of this other protein. It's a small monomer from the viroporin of hepatitis C. So in experiment, scientists have identified PS lipids as critical for the function of this ion channel and for the infectivity of the virus. And we just wanted to see if we have a membrane model with PS lipids versus without PS lipids, what changes in the protein-lipid interactions in this case. Our interest in P7, which is the viroporin from hepatitis C that we're studying, is because it's identified as a potential drug target from hepatitis C. I mentioned before, we're not looking at viral dynamics. Why I'm interested in P7 is because it assembles very close to where lipid droplets are formed. So lipid droplets are fat storages in our cells. They are particularly important in hepatocytes, in liver cells. Patients that have chronic infection of hepatitis C, they can live with it. They can have a very good quality of life or relatively good quality of life, but they are more prone to developing non-alcoholic fatty liver diseases or NAFLDs. If these are left untreated, this fat mismanagement known as steatosis can develop into fibrosis, cirrhosis, and sometimes that develops into hepatic carcinoma. And it all starts with fat mismanagement. So the question we're trying to answer is what's happening at the atomistic level, right, at the molecular level, with this ion channel from the virus that assembles very close to where the lipid droplets are formed, the fat storages are formed, that alters lipid metabolism in the cell. So P7 has been already linked through experimental studies to lipid metabolism, especially in the liver, and the management of fat in liver cells. So what is happening at the atomistic level in these protein-lipid interactions that result in something else happening with the lipid droplet? So our driving hypothesis is that as P7, which has 6 monomer forming the channel, as P7 assembles and forms the channel, it starts to change the local composition of the lipids around it in the endoplasmic reticulum, which directly impacts the lipids that are available to form lipid droplets. And we know that when the lipid droplets, and this has also been shown in experiment, or is currently being shown in experiment, when the lipid droplets have a different lipid composition in the monolayer, then they are either more difficult to digest, they are more stiff, maybe they cannot even leave the endoplasmic reticulum, and a whole cascade of events starts to unfold because these lipid droplets cannot serve as proper fat storages and energy sources for the cell. So again, our goal is starting from the simplest case of a monomer of the protein when it binds the membrane, how it changes the local lipid distribution, and in turn, the membrane properties of that local environment. That eventually, as you get more units of this protein and you form the channel and viruses, new viral particles are formed, everything is disrupted in the formation and digestion of these lipid storages or fat droplets, lipid droplets. So we started again with 2 membrane models. The main change between them was the addition of phosphatidylserine, identified in experiment as very important for the activity and the activity of P7 and infectivity of the virus. We had one control model without PS and a charge model with PS. And our findings were very interesting and encouraging. We saw that when you have PS lipids in the membrane model, the protein can actually insert into the membrane hydrophobic core. And when you don't have PS, the protein stays on the surface of the membrane. Again, these simulations were for nearly 2 microseconds. You will see on the plots that I'm showing 1 microsecond, data for 1 microsecond this is the data for when the protein was bound to the membrane. There's a tiny monomer. It did spend quite a bit of time in water until it finally bound, and then it either remained on the surface of our control model or was inserted inside the hydrophobic core of the charge model, which was the one with PS lipids. And then we started to quantify that behavior. We did hydrogen bonding, number of contacts, changes in the conformation and the structure of the protein, changes in the membrane. These are heat maps of the contacts between each amino acid in the monomer and different lipids in the membrane. You can see there's some enrichment of P lipids in the case of the control model. When you don't have PS lipids, you have a charge lipid, right? You have inositol lipids, but not phosphatidylserine, which seem to be or were suggested to be the key lipids for this protein. When you have PS lipids, then you don't have the enrichment of PE. You have, in fact, more PC lipids attracted to the protein and a few phosphatidylserine lipids. So that was for us surprising that PE, which is not that interesting, would be recruited in the absence of phosphatidylserine lipids. We also looked at the enthalpic contributions to the interaction energy between from each amino acid in the protein and the different membrane models. A few of them are conserved, right? A few of the highest contributors, G1, K3, this was lysine, but there are a couple that are present only, or there's one that is present only with phosphatidyllipids, phosphatidylserine lipids are present, and there is a tryptophan that is present in the interaction when PS is absent. So we were also seeing these markers of different interactions and a different lipid fingerprint when you have the protein interacting with different membrane models. And my student did dimensionality reduction analysis using TICA, or time lag independent component analysis, to try to identify, this was done on the dihedral angles on the helices in our monomer. And she was trying to identify which are the amino acids that contribute the most to these conformational changes in the protein as it interacts with different membrane models. So in the case of the control model, we see histidine 31 and tryptophan 30 as the main contributors to the different bound orientations and conformations of the protein. And you can see the 2D density map of the two major contributors to the TIC analysis. The map is very different. The heat map is very different from the two major contributors to the motions of the protein based on the same analysis when it interacts with the membrane model that has PS lipids. So when phosphatidylserine is present, phenylalanine 58 and N9 are the major contributors to the conformational changes in the protein. When phosphatidyl is not present, histidine 31 and tryptophan 30 are the major contributors to these changes in the protein conformation. The last thing I want to show you was the most surprising conclusion, or the most surprising result for us in this analysis. You can see here again the protein interacting with the control model and the protein interacting with the charge model. Phosphatidylserine present, phosphatidylserine absent. We were expecting to see differences in the lipid order of the inositol lipids or the serine lipids, which are the charged lipids in each of these models. However, the change was basically null in those cases. You can see very close to the membrane is the blue curve, and in the bulk region of the membrane was the red curve. Like the colors are a bit misleading with respect to the diagram here, but there was barely any change. When my student looked at the delta order parameter, or the delta SD, the most surprising results was seen with DPPE lipids. So in the control model, DPPE lipids are actually very disordered, right? You have splay angles between the tails very high. That's, you have less order there. You can see the red dots over here. But when phosphatidylserine phosphatidyl is present in the model, DPPE lipids near the protein they start to get very nicely in order with the tails parallel to each other. And you see their order parameters increase with respect to those in the bulk when they are very close to the membrane. So that was something surprising. We're still looking into that, why PE is having such a particular response near the protein when you have PS lipids versus when you don't have it. So the main takeaways from those studies were, again, that protein membrane interactions will vary depending on the membrane environment you reproduce in your simulations. We saw that when you have phosphatidylserine lipids, the protein inserts farther down into the membrane hydrophobic core. Histidine 1 and tryptophan 30 are very much important residues in this interaction and insertion mechanism. And again, something that we're looking farther into is the response of PE lipids in the membrane and how they interact with each other when the protein binds the membrane in the presence of presence or absence of phosphatidylserine in the membrane model. Those are the two case studies that I wanted to show you. That's a picture of my group when we were still wearing masks. I think I need to update that picture. But in any case, the main takeaway message of our studies is that protein-lipid interactions will result in a particular lipid fingerprint around the protein. And in the context of mechanisms of disease, we're trying to provide insights as to how these protein-lipid interactions are modulating the membrane response and reorganization of lipid domains and membrane properties, structural and mechanical, in the membrane. Thank you so much for your attention. I'll be happy to discuss any questions.
Thank you, Viviana. So that was a very interesting presentation. So now we are open for discussion. So I guess everybody should be able to unmute and turn on the camera also. If you have any question, please take a shot.
Yeah, can I ask a question? Hello, can you hear me?
Yes.
Yes.
Yes, it was a really nice talk, Viviana. So I'm Aravind, and I have a question regarding some of the observations you've done, especially on the PS and PE on the second part of the project.
Yes.
So have you seen when the PE lipid was recruited compared to PS, did you see any change, like what functional group was interacting in both cases? I guess the amine is present in both PS and PE, but since in this terminal, in the head region of PS, you have this carboxylic group as well. So I was curious if you look into that, like what functional group interacted with this protein and how would that affect the recruitment?
We did hydrogen bond analysis, and that is in the paper, but we have not yet looked at which specific hydrogen bonds there were. So we look at the occupancy of the hydrogen bonds, and we have the time series for that and some probability density distributions. But I think, yeah, we need to go back and check which portion of the specific amino acid is interacting, whether with the phosphate region or with the carboxylic head group. Actually, that's something that we need to check. Other than the specific amino acids interacting with it, I don't have an answer for that yet. So it's the second helix in the protein is the one that is interacting the most with PE lipids. My guess would be just simply looking at the pictures, right? This is the second helix, this white one, or mostly white one here in the slide. So when it's interacting with the lipids, I would say it's mostly with the head group and not the phosphate, right? It loses that interaction when it goes in, right? So the second one it's in and it's not interacting with PE. I have to check though. I will make sure to tell my student to look at which is the specific hydrogen bond interacting with it. Thank you. I'll write it down before I forget.
I think I would have a question.
Yes.
So so you were mentioning actually that there are some mechanical properties that the lipid, once the fingerprint, the interaction between the protein and the lipid happens, and then a fingerprint
You're cutting off. I heard until the fingerprint is generated, and then that was, I couldn't hear anymore. Can you repeat the question, Adolfo?
Yes. Look,
There you go. Now I can hear you.
Yeah. So the question is about the mechanical properties of the membrane. So
Yes.
in your studies, you made the interaction between the lipids and the proteins, and then you have the fingerprints. So, okay, I understand it's more like some kind of heterogeneity in the membrane that actually occurs under the presence of a protein. But in terms of the mechanical properties, how this is an extensive property of the full membrane. So it's something more like a stiffness or some kind of or mechanical parameter that defines the membrane. So are the fingerprints somehow triggers of changes at the level of the stiffness of the entire membrane, or how do you see this issue of mechanical properties?
Because in the case of both proteins, right, they are fairly small compared to the membrane. And especially in the one that I'm showing right now on the slide, which is the MLKL, we know it most likely doesn't act just one protein at a time. So some people suggest it's a trimer, or the people say it's a tetramer. So we are hoping in the future to look at more units. My student is trying to do some pseudo docking to see how the protein-protein interactions work, and then he will put them on the membrane. But we look at, for now, we're looking at the changes in the properties very close to the membrane. Then we define a buffer region, and then we look at the bulk. So we typically start with the order parameters, and we try to see the extent of the effect of the protein on the membrane, right? So in this case, we start to look, let's say 3 angstroms within the protein, or within the heavy atoms of the protein. What is the order parameter? And then 5 angstroms, and then 7, and then 10. And we define that way a radius of impact of this protein. And then that's what we use as our local environment. And then everything else minus a buffer region, everything else is the membrane bulk. So for now, we're just trying to look at these local changes. And then as we start to include more protein units, then I think we can start to look maybe at the line tension, or maybe pressure profiles of the entire membrane, to see how these local changes further affect whatever is happening far away from the protein.
Okay. Okay, thank you.
Thanks. Any other questions?
Yes, anybody else? I mean, I will have one very brief, but it is related to the use of Anton. So
Yeah.
you use Anton. I mean, Anton has a specific, I mean, it's a computer that can speed up molecular dynamic simulation, basically. And so the time scales of microseconds and under 200,000 atoms, is it still necessary for Anton, or is for a very large scale simulations? I mean, the ones you show.
Current, right, the current Anton, so Anton 2 can handle up to 700,000 atoms, which is the size of these simulations of the MLKL and the membrane. In our cluster, we can run that 2 microseconds, maybe 2 months. In Anton, we run it in 20 hours. So it's 100 times faster. Yeah. Now, when you apply for Anton, one thing is the size, but they also, so the committee that evaluates it also sees the necessity. So in our case, we justify it because we have complex membrane models. So we want to allow for that lipid diffusion to equilibrate, and then we have to run at least 1 microsecond or 2 microseconds, right? So probably if we were to send the small monomer for P7 and the membrane, maybe you won't carry as much weight because those are smaller membrane patches. We can run it in our computer. But for these ones, to get it in a day instead of 2 months is much better.
Yeah, so you are telling me 2000 nanoseconds in a day. So yeah, this is
Yeah.
way.
It's fun. Now, you only get you can get up to 8 days on the Anton machine. I've used it in the past when I was a grad student and a postdoc, but now as a group leader, then we're getting 4 days because we're a junior group. And then as we start to publish more of the material and establish ourselves, then I'm hoping we'll get the full 8 days.
Oh, nice.
Then the challenge is the analysis after that, right?
Yeah,
because we have so much data, and then trying to do time correlations, dimensionality reduction, which is actually the side on method development that I envisioned my group getting into, not really in the molecular stimulation, but mostly on the analysis. But we'll see. It's we're starting to study the P7 monomer, or we want to study with monolayers for lipid droplets, because we have a collaborator who can study the lipid droplet properties. So even the setup for that is proven a little bit challenging. So we'll see where research takes us.
Yeah, actually, I was thinking also the amount of data that you generate. So that I mean, that's another issue, right? So
yeah,
years ago, I mean, we were thinking that actually core screening could be used also as a tool to, to filter that amount of data. So the other way around. So nowadays, we use core screening to mimic all atom simulations, but actually it can be used also as a modern tool to filter data, more or less like you do TICA or you do PCA or something like that. But I haven't heard more about those ideas recently.
But those when I started using them, which was later in my postdoc, so PCA, TICA, I wasn't using those before because also my proteins were very small, like 27 amino acids or something. So then I started to look at bigger proteins, and they don't always give you the insight you want, right? So in this case, so she was able to get this using dihedrals and TICA. So you have this nice step change in the TIC coordinate. But we have tried that. This is what, 67 amino acids or something like that. We try it in the bigger protein, in the MLKL, and we get something more like this one, right? It's more smooth. So we don't have a good descriptor for the analysis. So my student was saying, maybe TIC and PCA don't work, and he was trying to do some VAE variability, something. I don't know. He's looking into these fancy approaches, and I'm learning with them, which is the exciting part. Right? They bring new insights. I'm very fortunate that my students have a background, one of them in biology, the other one in physics, the other one in math. So all this is cultural and academic diversity it's a real asset in the field.
Definitely. Okay, so coming back to, there are some messages. Let me see, what is this?
Is there time for another question?
Yes.
It's more of a technical question to ask. So the first one is like a little more technical in terms of what I may have missed it in the talk. So what was the force where you used? And the other thing is, so first the HMR used to improve the time scale. And do you have any comments on usage of HMR for lipid simulation? I'm quite new to lipid simulation, so I'm still trying to figure out here and there. So just want to get your thoughts on that.
Thank you for the question. So the communication was a little bit broken, but I think one of the questions was the force field I use, and the second one, hydrogen mass repartitioning.
Exactly, yeah. And especially because there was a paper recently that if I recall it correctly, about the ligand binding, where the diffusion was disfavored the ligand binding for the protein substrate, if I recall it correctly. So I'm wondering whether you observe anything in terms of lateral diffusion of it because of the HMR or without.
So with HMR, you're sort of rescaling that vibrational frequency of the hydrogens, right? So instead of, which is why you can run at a higher time step. So typically you run 1 femtosecond, 2 femtoseconds, and then you use hydrogen mass repartitioning at the atom level, you can go to 4, maybe 5. So what you're doing is, if you imagine this, if this is the curve that you're trying to fit with the numerical integration, you get a bit more error as you integrate at a higher time step. So I think if you just do membrane only, it may go better. You still, I would still do some benchmarks, do all atom 2 femtoseconds, and then all atom hydrogen mass repartitioning at 4, and see that it reproduces the properties I'm trying to study. I am interested in the specific interactions as the question that was asked earlier, what portion of the lipid is interacting with what amino acid? If I have hydrogen mass repartitioning, I would probably have higher uncertainty with that, but I'm interested in that specific interaction. So we run brute force MD, all atom simulations, charm 36M for the force field of proteins and lipids. But yeah, I think for membrane only simulations, it may go better. Or if you just have a massive protein and you're trying to understand the basic motions of the protein, always do a benchmark. I will compare that, the all atom versus the HMR, and see that it reproduces my radial distribution function. And if it does, then use that one for that particular study.
Thank you.
There is a question in the chat. Actually, is elaborated by Wisha Lu. I don't know, maybe you want to ask if you can unmute.
I'll see the question in the chat.
But basically, it says in page 24, how do you define the context between the HT9 and the lipid molecules?
I think it was this one.
Yeah.
We basically count. So we looked at 1000 nanoseconds of bound trajectory. So the simulations I mentioned before were close to 2 microseconds each, but we look at one for all of the replicas we had, which were 6 in this case. We look at the bound trajectory of the protein, and then for each alpha carbon of the individual amino acid, we define a radius of 5 angstroms, 5 or 7 angstroms, and then we count how many lipids of one species or another are around it, or how the phosphate, really, the phosphate atom, which are these spheres. Between the alpha carbon and the phosphate atom of the lipid, we count in those 1000 nanoseconds, how many contacts are there, and then we do the analysis. It's just counting, really. Just distance, a cutoff radius, and just counting the number of phospholipids of PC, PE, PI, and the hydroxyl atoms of cholesterol.
Okay, so okay, thanks a lot. Thanks.
Thank you. Thank you for the questions.
Anybody else? Okay, we are just going over 4. So here, basically, we will say you thank you, Viviana. So
yes, my
to have
pleasure.
you here. And so just a quick announcement, in 2 weeks, we will keep the seminar. So the next speaker will be Professor Jan Bresowski. So he's a local speaker, well, not from Warsaw, but very close by city, Poznan. He will talk about computer analysis and design of ligand transport processes critical for protein functions. So it is still within this membrane. So maybe you will be also interested in joining, but unfortunately it will be a regular midday.
Right. But how many hours are you away? 6, 7? I have 10 a.m. for me, right?
10. Now it's 16. 16.
So
6 hours.
6 hours. Yeah.
Yeah. It's kind of the drawback with it already happened with our first speaker. So, but anyway, so you made it, and thank you
so much for the invitation. I enjoy meeting everybody else. Thank you, Adolfo.
Take care, Viviana.
You too. Thank you. Thanks, everyone. Have a good rest of the day.
You too. Bye-bye, guys.